15% of the human population carries blood that science cannot trace to any known origin on Earth. Not a disease, not a disorder, just blood, sitting in veins, keeping people alive, defying every serious attempt to explain where it actually came from. And the deeper researchers look, the stranger it gets. 85% of humanity shares a biological protein that connects them through an unbroken chain to the primate family tree. The science on that is clean, consistent, and settled. But the remaining 15% are missing that protein entirely. And when researchers follow that absence backward through the genetic record, looking for the source, looking for the ancestor, looking for the moment this blood first appeared, they hit a wall. No primate link, no clear origin, no laboratory on Earth has ever successfully reproduced it. For decades, the working explanation was mutation of unknown origin. Scientists named it, filed it, and largely moved on. But new advances in ancient DNA research are finally cracking open what that origin might actually be. What they found was not what anyone expected, and the answer has implications that stretch far beyond medicine, far beyond genetics, and deep into a question that human beings have been asking about themselves for as long as they have been capable of asking questions at all. To understand why Rh-negative blood is so extraordinary, you first need to understand what the Rh factor actually is, because the mystery only becomes visible once you see what science considers normal. Coating the surface of your red blood cells, there are proteins, dozens of them. They function like biological identification tags, markers your immune system uses to distinguish what belongs from what does not. One of the most significant is called the rhesus factor. Named after the rhesus monkey where it was first identified, this protein is ancient. It predates modern humans by millions of years. It runs through the entire primate family tree without a single interruption. If you are Rh positive, this protein is present. Your immune system recognizes it as self. It has always been there, written into your biology exactly as it was written into every primate ancestor before you. Rh-positive blood comes in 4 variations, A, B, AB, and O, but all of them share this one defining characteristic. The rhesus protein is present. The evolutionary lineage is intact. 85% of every living human being falls into this category. That number matters. In biology, when 85% of a species shares an ancient inherited protein connecting them to a clear evolutionary line, that is not coincidence. That is the signature of deep time. That is genetics behaving exactly the way evolution predicts it should, which is precisely why the remaining 15% is so difficult to explain. Rh-negative blood is not a lesser version of Rh-positive, not diluted, not damaged. It is the complete and total absence of the rhesus protein from the surface of red blood cells. That implications are anything but. Every other known primate on Earth carries the rhesus factor. Chimpanzees, gorillas, bonobos, orangutans, the entire evolutionary family that modern genetics has mapped across decades of research. The protein is there consistently across millions of years of biological history. When scientists trace Rh-positive blood backward through the genetic record, the lineage is clean and unbroken. It connects. It makes sense. Rh-negative blood does not connect. Follow it backward, and the trail goes cold. No primate ancestor carries this variant. No gradual mutation is visible in the fossil record. No moment in the evolutionary timeline where a clear before and after can be identified. It simply appears. A few thousand years ago, in geological terms, practically yesterday, this bloodline emerged in the human record without a traceable biological parent. Scientists have proposed that Rh-negative is the result of a mutation. That is the current working hypothesis. But here is what that explanation quietly glosses over. A mutation of what exactly? Mutations require a source. They are changes to something that already exists. Yet the something that Rh-negative supposedly mutated from has never been identified. Laboratories worldwide have successfully synthesized proteins found in Rh-positive blood. They cannot do the same for Rh-negative. There is no protein to reverse engineer, no template to copy, no ancestral form sitting somewhere in the genetic record waiting to be found. Science knows what it is. It still cannot explain where it came from. And within the Rh-negative group, there is one type that stands apart from everything else. O-negative blood. Less than 7% of the global population carries it. Some estimates place the number closer to 5%. In a world of 8 billion people, that is an extraordinarily small group holding something the rest of humanity depends on without ever thinking about it. Here is what makes O-negative unlike anything else in the blood type system. It is the universal donor. When someone arrives at a hospital unconscious, bleeding, with no medical history and no time to run tests, O-negative blood is what goes into their veins. It requires no matching. It triggers no immune rejection. It crosses every biological boundary that blood type compatibility normally creates. A positive, B negative, AB positive, O positive, it does not matter. O-negative works for all of them, the rarest blood on Earth, and the only blood that can save anyone, regardless of who they are. But the paradox cuts the other direction just as sharply. O-negative individuals cannot receive blood freely in return. They can only accept O-negative. Every other blood type, even other Rh-negative variants, is incompatible. The body rejects it. The immune system treats it as invasion, the blood that everyone can receive from almost no one. That asymmetry is not just medically fascinating. It is biologically strange in a way that evolution alone struggles to account for. Think about the design of it. The rarest variant gives freely to everyone. Everyone else gives nothing back. In any standard evolutionary model, a trait that is costly, one that asks everything and returns nothing in material terms, should not survive. Natural selection does not favor one-way generosity. Yet here it is, persistent and stable across thousands of years of human history. A trait this specific, this rare, and this functionally extraordinary does not appear randomly. It appears with a history, and that history is only now beginning to come into focus. Because Rh-negative blood does not appear randomly across the human population. If it did, that would actually make it easier to explain. Random distribution would suggest a mutation spreading gradually and evenly through interbreeding populations over time. That is not what the data shows. It clusters. It concentrates in specific regions among specific populations in patterns too consistent and too geographically precise to dismiss. And when you map those concentrations onto the history of human movement across the planet, something remarkable begins to take shape. The highest known concentration of Rh-negative blood in the world is found among the Basque people of northern Spain and southern France. Studies have recorded Rh-negative rates in this population exceeding 30%. That is not a slight statistical elevation. That is a number so far above the global average that it demands an explanation. The Basques are not just notable for their blood. Their language, Euskara, has no known relationship to any other language on Earth. Their genetic profile is distinct from every surrounding population in ways suggesting they have remained relatively unmixed for an extraordinary length of time. Their culture, their ancestry, their very genetics mark them as separate from every neighboring group in Europe in ways that researchers have never fully resolved. They are, in the most literal biological sense, a people apart. Beyond the Basques, elevated Rh-negative rates appear consistently in Ireland, Scotland, England, parts of Scandinavia, the Atlas Mountain regions of North Africa, and certain populations across the ancient Middle East. In contrast, Rh-negative blood is exceptionally rare in East Asian populations and extremely uncommon across most of sub-Saharan Africa. This is not a random scatter, this is a map. And maps have origins. They point somewhere. They point to a time, a place, and a people that this blood came from, and to a question that has been sitting unanswered for as long as researchers have known where to look. For most of human history, the tools to trace that origin simply did not exist. Researchers could observe the distribution, they could document the mystery, but the genetic technology required to follow a bloodline back through thousands of years of human prehistory was not available. That has changed. Over the past 2 decades, advances in ancient DNA extraction have allowed scientists to recover and analyze genetic material from human remains tens of thousands of years old. Bones, teeth, fragments preserved in permafrost or cave sediment are now readable in ways that would have seemed impossible a generation ago. The science of ancient genomics has accelerated faster in the last 10 years than in the previous century combined. And what that technology is revealing about Rh-negative blood is beginning to rewrite the timeline. Current findings point toward a specific and surprisingly narrow origin. The genetic variants associated with Rh-negative blood trace back to a small founding population concentrated in Western Europe and parts of the ancient Middle East that predates the major migration waves which shaped the modern human map. This was not a widespread mutation rippling across the entire species. It was a localized event in a small group that then traveled through specific lineages rather than dispersing broadly. Researchers examining ancient burial sites across Western Europe have found Rh-negative markers appearing consistently in remains dating back further than previous estimates suggested. The picture that emerges is not of a bloodline that appeared recently and spread quickly. It is of a bloodline that is very old, very contained, and in many respects very deliberate, as though it moved through history along its own distinct path rather than blending into the broader human story around it. That explains the clustering. It explains the Basques. It explains why the geographical distribution looks less like a random biological accident and more like the echo of an ancient journey taken by a very specific group of people whose descendants are still here, still carrying the same blood their ancestors carried thousands of years ago. But it does not explain why this bloodline stayed rare. That answer comes from somewhere far more intimate and far more brutal. When an Rh-negative mother carries an Rh-positive baby, her immune system can identify the rhesus protein in the fetal blood as foreign and begin producing antibodies against it. In a first pregnancy, the risk is manageable, but in subsequent pregnancies, those antibodies cross the placenta and attack the child. Before modern medicine understood this process, it killed children repeatedly, constrained family sizes generation after generation, and created a powerful ceiling on how far Rh-negative genes could spread through any population. The bloodline was not just rare, it was actively suppressed by its own biology. For millennia, that suppression quietly shaped which lineages survived and which were narrowed. It was not a wall, it was something slower and more relentless. A filter, operating across thousands of years, generation by generation, tightening around every family that carried this blood. In practical terms, this means that every Rh-negative person alive today is descended from ancestors who managed, against significant biological odds, to pass this bloodline forward. The families that carried it were not just rare, they were survivors of a reproductive gauntlet that eliminated most of the others. Whatever this bloodline is, the people carrying it today come from a very long line of people who refuse to disappear. That ceiling was removed in the 1960s with the development of the ROGAM injection. For the first time in human history, Rh-negative genes can propagate without that ancient biological constraint. The filter that spent millennia shaping this bloodline was switched off within a single generation. The implications of that are only beginning to unfold. But here is where it stops being purely a story about blood. Beyond the medical data, beyond the geographical mapping, beyond the ancient DNA timelines, there is a pattern that researchers and communities of Rh-negative individuals have been documenting for decades. A clustering of physical and cognitive traits that appears consistently enough across independent observations that dismissing it entirely has become increasingly difficult to justify. The physical characteristics reported most frequently are specific. Higher rates of blue, green, and hazel eyes, lower average blood pressure, heightened sensitivity in both vision and hearing, a body calibrated differently, running quieter and registering more. The cognitive and psychological profile is where it gets harder to explain away. Higher measured IQ scores appear in multiple independent studies. Strong empathic sensitivity, a heightened ability to recognize patterns that others miss, a disproportionate pull toward questions that mainstream thinking has not yet answered, the unexplained, the anomalous, the edges of what is currently known. That last point is worth sitting with, because it is not describing intelligence in the conventional sense. It is describing a specific orientation, a mind that moves toward the unresolved rather than away from it. That finds the incomplete answer more interesting than the settled one. And then there is something that no blood test can measure, but that surfaces with remarkable consistency across cultures, geographies, and generations of Rh-negative individuals. A feeling of not quite belonging, not dissatisfied, not dysfunctional, a persistent quiet sense of observing the world from a slight distance that others do not seem to feel. Science has not yet established definitive causal links between Rh-negative blood and these traits. The honest position is that the associations are observational and the mechanisms are not yet fully understood. But observational patterns that replicate independently across thousands of unconnected people are not nothing. They are the beginning of a question worth taking seriously. If a single ancient mutation can reshape immune response, blood chemistry, and reproductive compatibility across thousands of years, the idea that it left everything else completely untouched requires more faith in coincidence than the data currently supports. The geographical clustering is real. The reproductive barrier that kept this bloodline rare for millennia is documented. The ancient DNA findings are pointing toward an origin that predates everything previously assumed. And the traits reported consistently across Rh-negative populations worldwide are too persistent to keep ignoring. What ancient DNA has revealed is not a final answer. It is a more precise question. And in science, a more precise question is always progress. For most of human history, this bloodline was invisible to the tools available to study it. Researchers could see its effects, map its geography, and document its medical behavior. But the underlying story, the origin, the timeline, the chain of events that produced it, was locked away in material that no one yet knew how to read. That is no longer true. Consider what we now know. A small founding population, ancient and geographically specific, carried a bloodline that no other primate shares. A biological mechanism spent thousands of years suppressing that lineage, eliminating children, narrowing families, quietly culling the bloodline across countless generations. And then, in 1968, that mechanism was switched off entirely. Whatever this bloodline is, wherever it came from, it is now, for the first time in its entire existence, free to move through the human population without limit. The story is not closing, it is opening. And what comes next will force questions about human origins that the previous century of science was never equipped to answer. Before we start, what part of the world are you in? Drop a comment below and don't forget to subscribe.